Good morning, everyone. Welcome to Grand Rounds. Welcome to those of you who have joined us in person and those who are joining us on the Zoom webinar. Just want to make sure. Okay, good. Audio is good on the webinar. We're really pleased to have you join us. I have a few announcements before we introduce today's speaker. So, and I'm Jeff Miller. I'm joined remotely via Zoom by Christine Denny today, one of our co-directors of Grand Rounds, and Kate Elkington is our third co-director. So first, an announcement about next week, about scheduling. Next week's Grand Rounds will be in person as well. It will be part of the Addressing Structural Racism series, organized by the Office of Equity, Diversity, and Inclusion. The speaker will be Dr. Sean Joe, MSW, PhD, founding director of the Race and Opportunity Lab at the Brown School of Social Work at WashU in St. Louis. Great, and as I said, next week's will be an in-person Grand Rounds like today's. Feel free to come back and join us for lunch, both at the conclusion of today's session and at the conclusion of next week's session. Okay, and then a few comments about questions, the Q&A at the end of today's session. We'll welcome questions from the in-person audience and also from our Zoom webinar attendees. We really try to prioritize trainee questions, both from the in-person and the Zoom audience. The way that you can ask a question if you're joining us via Zoom is by posting your question in the Q&A function at the bottom of the Zoom window, not in the chat. If you're a trainee, please put the word trainee at the beginning of the question. And then for Zoom webinar attendees, we would really love it if you would ask your question yourself. To do so, we would temporarily promote you to a panelist so that you could ask the question camera on or off according to your preference. So if you post a question in the Q&A, please write at the beginning of it, can ask question myself or prefer to have my question read. Okay, I'm very pleased to introduce today's speaker, Dr. Tarjinder T.J. Singh, who has several current roles, assistant professor in computational and statistical genomics in our department, with an interdisciplinary appointment at the Mortimer B. Zuckerman Mind-Brain Behavior Institute. He is also associate faculty member at the New York Genome Center. And our department was thrilled when we were successful in recruiting Dr. Singh to our faculty just this past summer. I believe August, TJ, is when you joined our department. And so it's really great to welcome him to Grand Rounds to share his work with us, hopefully to foster collaboration and to meet more of our department in person. Okay. Dr. Singh received his bachelor's degree in biology, mathematics, and economics at Williams College. He completed graduate studies at the University of Cambridge and the Wellcome Trust Sanger Institute with Dr. Jeffrey Barrett in 2016. He continued his training at the Analytical and Translational Unit of MGH, the Mass General Hospital, and the Stanley Center for Psychiatric Research at the Broad Institute of Harvard and MIT with Dr. Mark Daly and Dr. Benjamin Neal as a postdoctoral fellow and an instructor at Harvard Medical School. As part of global collaborative efforts, he has analyzed large-scale sequence data to identify protein coding variants in individual genes that confer substantial risk for psychiatric disorders. He's interested in using genome sequencing as a foundational tool to understand the etiology of mental illnesses. And although he didn't put this in his biography, I will add that he has published important work on the genetics of schizophrenia, epilepsy, and autism spectrum disorders in high-impact journals, including Nature, Nature Genetics, and Nature Neuroscience. So we're really thrilled to welcome Dr. Singh today. The title of his talk is Insights into the Genetic Architecture of Psychiatric Disorders through Population Scale Sequencing. Welcome, TJ.
Thank you for that kind introduction, and it's great to be here and introduce myself to this community. I've started at Columbia a couple of months ago now, and it's been a blast working with you at the Department of Psychiatry, meeting other faculty and researchers at the Zuckerman, as well as working with many colleagues at the New York Genome Center. And so today I want to give an overview of 2 vignettes of work that give some insights into what we've learned now with genetics in the past 20 years. And with the last bit of the presentation, I will highlight areas of interest and exploration of what we will do, hopefully, in the next couple of years to further push along the area of genomics in psychiatry. And so I guess we should share screen and let's get going. Something is happening on my screen, so I'm just going to let it happen. Great. Okay, so insights into the genetic architecture of psychiatric disorders through population scale sequencing. So why is genetics important in medicine? Well, a lot of conditions, traits, diseases, all have a genetic component. And one of the key questions is why particular individuals have higher risk for specific diseases and outcomes. And given there's a genetic component, some heritable component, we can then use technologies to find precise genetic markers and genes that confer risk for these conditions. And then through these clues, we can gain insights into the mechanism and the biology of disease. And so genetics, in a way, for genetic disorders, we can get a lot of clues that move us forward in understanding etiology, biology, and hopefully better treatments. And I think, we show this plot a lot when we think about psychiatry. And on the y-axis there is what we call heritability, the genetic component of traits. And one being fully heritable, high genetic kind of heritability, and 0 being not heritable at all. And on the x-axis are different disorders that we look at in psychiatry. And there are 2 different measures. There's the H-squared, the first one, which is broad sense heritability, looking at twins and families, and the second one using genetic markers. And what is clear is that behavioral traits and neuropsychiatric traits are amongst the most heritable of all disorders and behaviors in humans. And so we see more severe disorders, maybe schizophrenia, bipolar, and then there's a spectrum where there's more complexity, perhaps, less severity, and the genetic component like depression is a bit lower, but still heavily substantial. And one thing I want to also note is that none of these are one. And so all traits have a genetic component, environmental component, and chance. And so genetics is one of many angles for which we could understand psychiatric disorders. But it is an important one because these traits are substantially heritable. And so how do we study the genetics of traits? Well, this plot here is a bit complicated, but crucial to understanding what we call the genetic architecture. On the x-axis there is what we call allele frequency, how common a genetic marker is in a population. So a common marker would be 1 in 100, 1 in 20. So many of the people here would carry that marker. And because it is common, it cannot by itself cause schizophrenia, for instance, because otherwise the incidence wouldn't match what we know, which is 1%. And now these have been incredibly hard to study for the longest time in history because their effects are so small in the population. On the left-hand side are really rare variants, very rare genetic variants, which individually can confer substantial risk, but they're rare in the population. And we've been stuck in a century of genetics of how to measure these. We didn't have a reference genome. We didn't have technologies to measure it. And the first glimpses of these required very specific study designs. In the 1980s, genes on cystic fibrosis, Huntington's disease, we could follow markers over large families to study it, but didn't work for psychiatry because it's not that simple. It's not simple, but psychiatry is another level of complexity. And so for a century, we know there's a genetic component, but we didn't have the ability to find these markers. And so it took really the turn of the century, the initial sequencing of the human genome, to initially map out where the genes are, where the markers are, to begin the process of gene mapping. That was completed in 2003. And then what's the point of one genome when you have to study variation, differences and similarities between peoples? And that required the HapMap project, which looked at differences in a couple of hundred people to begin the process of finding common genetic markers. But then it wasn't very diverse and the numbers are too small. Then it went to the 1,000 Genomes project, which is at 2010, and it finished maybe in 2015 or so. We looked at 1,000 genomes around the world, which is more representative. And now, after that, we're now in the tens of thousands, hundreds of thousands, and emerging millions of genomes. All of this happened in the last 20 years, where the democratization of genotyping and sequencing is allowing us to study many traits and many diseases. And so I'm new, I think I was heavily shaped by these events in the last, 10 years, I guess. And I think, we talked about my journey. I was a broad liberal arts student, but then I got the chance to go to the Sanger Institute at the University of Cambridge, where a third of the human genome was sequenced. And I saw, this is, early 2010s, how these technologies were scaled up for gene mapping and understanding of diseases. And I continued my training in Boston at the Broad Institute, which is kind of it came out of the Whitehead Institute, where another third of the human genome was sequenced. And so I think my thinking and my approach, and hopefully what I'll bring to the department, is this angle of genetic technologies as applied to medicine. And so now I'm going to give 2 vignettes focused on schizophrenia and psychosis, and kind of the insights we've gained from a large meta-analysis of analyzing tens of thousands of people with schizophrenia, and then focusing on a population in Finland, where we have deeper biobank data, and we can learn from that kind of approach. And so I'm not going to explain schizophrenia to this group, but just to emphasize, there's a strong genetic component, and there's been little progress in developing new therapeutic targets because the etiology, the biological processes towards this understanding this disorder is still very, there's a lot to be learned. And because of that, unlike cardiovascular disorders, other disorders where the pathways are better understood, we're kind of hitting a wall in finding a lot of new targets. Now, I mentioned that there's been a lot of genetic studies in the past decade, and schizophrenia actually is one of the best examples of how this transformed, I think, our view of the whole field of statistical genetics. So here we have a Manhattan plot. On the x-axis are the human chromosomes. On the y-axis are transformed p-values, how associated it is to schizophrenia. And we now have markers across the board that are linked to schizophrenia. This took, a long time. But now in 2022, we now have 300 loci that are linked to schizophrenia, which is remarkable. However, our goal is to understand biology and disease. And when we zoom into one of these markers, what we see is that it is unclear a couple of things. What the causal variant is, because of cystics, we actually don't know which one is the real variant. There's a correlation of variation. And given that variant, we don't know which gene it is. So it becomes very hard to understand the biology. And if you look at the vast majority of these GWAS common variant associations in schizophrenia, only a couple are coding variants. And so we have a massive task to go from these 300 hits, which are regulatory variants, mapping to unknown genes really, into pathway and processes linked to schizophrenia. And so let's go back to that plot I mentioned earlier. Frequency in the population on the x-axis, effect size, kind of predictability, penetrance on the y-axis. And so a lot of studies have now kind of together found these common variation, 300, that confer a risk for schizophrenia. Their effects are small because they're common. They cannot cause schizophrenia, but there's a lot of them. And if you add them up, they actually explain a lot of the genetic component for schizophrenia, but they're hard to study. Now, on the left-hand side, what we have there are these rare variants. They each can give you a lot of risk for schizophrenia, but these are harder to study because they are rare. You have to study a lot of people before you see them. Now, a lot of amazing scientists focused on copy number variants, structural variation. These affect large parts of the genome. 3 million letters at once, dozens of genes. One of them, which you probably know well here, is a 22q11 deletion. And so that one could be mapped out because you can actually visibly map it out pretty easily. And there's about 11 of those. But these hit a large number of genes. And so we can use new technologies, sequencing technologies, to pinpoint specific genes and link them to schizophrenia. And so that's where whole genome sequencing, whole exome sequencing comes in. It could fill in that gap and find genes in which large effect protein variants, a stop codon or a frameshift mutation, give you 50-fold higher risk of schizophrenia, 20-fold higher risk. And that's where I kind of begin my journey into genetics. And just to show you what rare means, here is a transform plot. It's log transform. And so when I mean rare, this is a uniform transformation. It means really rare. So 1 in 10,000, 1 in 100,000. That's the frequency for which we need to find these rare protein coding variants. And so the overall message is that as we could do common variant studies, now with the decreased cost of sequencing, we can look at large amounts of risk genes, look at rare but high impact coding variants to find genes. We can model those with greater ease than regulatory variants. So a lot of the basic neuroscience work are gene focused. Animal models, knockouts. With these genes, we could leverage those models and understand biology a lot more easily. The problem is, as I mentioned, they're really rare due to natural selection. And so we have to sequence tens of thousands of people before we find these markers. And so I mentioned 2 scientific vignettes. The first one is insights from large-scale genome sequencing and what we learned from that. And the second is how we could build patient cohorts and deeply phenotype these patients to learn more about the psychosis spectrum. And then at the final bit, we'll talk about things that we'll do in the future. And so what we see as cost of sequencing decreased is that we generated a lot more sequencing data in schizophrenia and controls. In 2010, a couple of hundred, not super exciting. We didn't find much. Well, I wasn't there, but like people didn't find much. And then in 2013, about 7,000 were sequenced in the global kind of community and over 10,000 controls. And this led to an avalanche of papers. The first ones are in 2014, which proved that there were rare variants in schizophrenia that conferred risk, but we couldn't find individual ones because we didn't have enough people sequenced. And then I began some of my work as a graduate student and postdoc, where we mapped out initial glimpses of genes and also patterns with different psychiatric presentations, but it was still a hint of what is to come. And so in 2017, a group of investigators from around the world formed the Schizophrenia EXO Meta-Analysis Consortium, which put together all the sequencing data for neuropsychiatry in a bucket, and we kind of analyzed it as a community. And through that, it reached over 25,000 cases in the first analysis and over 50,000 controls. Now, this was truly a global effort with hundreds of investigators and scientists, and we really see this in the intention of recruitment and scale. So when I first started, I was in the UK, and the red dots and blue dots are the patients and controls in schizophrenia. As you can tell, they're not very diverse. They're all from England, Finland, Sweden. You know, we learned stuff, but it was not what we wanted to do in genetics. And so with the schema analysis, through work of kind of leaders like Thomas Lehner here, they funded a lot of studies that led to more diverse populations. And that is a good start, but something we need to continue to commit to as we generate more genetic data in the future. And so I'm not going to go into a lot of the methodological detail, but to say that we're doing a genetic study comparing rates of mutations in cases compared to controls, 24,000 cases and 100,000 controls. We're looking at ultra rare variants. Variants are seen in 5 in 50,000 people or 1 in 50,000 people. So these are really rare variants. And they're usually stop codons, frame shift mutations, really bad variants that affect the function of a gene that leads to haploinsufficiency. We focused on case control design, so the 24,000 and 100,000 controls. We also had families, 3,400 trios, parents and their child, to focus on de novo mutations, new mutations in the population that have not gone through a lot of selection, as we call it. And we combined all this data for an initial effort for gene discovery. And what we found, so actually, this is a good example. SETD1A, you might have also heard about this gene before. It is a chromatin modifier. Loss of function variance in SETD1A increases risk for schizophrenia. What does this look like in the schema analysis? We see 3 de novo mutations that are not seen in the parents, it's seen in only the children. We see 15 loss of function variants in the cases, 3 in the controls, and this is a 1-to-4 case control ratio. and we see signal and missense variance as well. And if you combine all that evidence using different statistical methods, this becomes one of the top genes that are linked to schizophrenia. And this work initially identified 10 genes that are conferring substantial risk for schizophrenia, where each one of these could lead to 3 to 50-fold higher risk. And what is also clear is that there's a long tail of genes to come. There's likely hundreds of genes for schizophrenia, and we think we have the first 10, 30 of them, and is a beginning of a trail of discovery for this trait. And just to put it into context, this is the plot that I showed earlier. Common variance, small effect on the right-hand side. And now we have the copy number variance, 22q, other large deletions and duplications, and now single genes that confer substantial risk for schizophrenia. Some of them are pretty remarkable. If you take exportin 7, nuclear transporter, we don't really know what it does, 50-fold higher risk of schizophrenia. So 1% chance of getting schizophrenia, 50% chance if you have that variant. And so we're now finding a class of variants that have individual predictive ability in a small number of patients. And I'll explain that a little bit more as I go on this talk. So insights, what do we learn from this kind of work? And I think the first bit is biological insights into the etiology of schizophrenia. And so we think a lot about pathways and processes. We can aggregate and look at all the known processes that have been studied by different investigators, gene ontology, KEGG, if you know these kind of pathways, and we tested every single one to see if they're linked to schizophrenia, to see if they carry genetic signal. And we found a long tail of pathways for which there is an enrichment. We tested 4,000, and we saw 50 or so that had really strong enrichment for schizophrenia. And I'm not going to make you look at these small letters, but to say from the genetic data, the list from before all hits these groups. They're genes for the postsynaptic density, synaptic transmission, ion transmembrane transport, voltage-gated cation channels, axon guidance. So they all hit pathways and processes we suspected to be linked to schizophrenia through a lot of other studies, but they come naturally from the genetic data. What we also did was took expression data, human tissue, and we measured their gene expression, and we saw how much of that overlap with the genetic data as well. And not very surprising, the top ones are all brain-related tissues. From the prefrontal cortex to the cerebellum, you can see a gradient of different brain tissues and nothing in the rest of the body, which is also reassuring of what we know about schizophrenia. And then we also see where it is in single cells. And I think there's going to be a lot more work in the near future about genetic enrichment in multi-omi and single cell data. When we look at neurons and non-neurons, glial astrocyte cells, all the signal is in CNS neurons, nothing else anywhere else in the peripheral nervous system, no genetic enrichment. And then when we look at excitatory and inhibitory neurons, we see a couple of brain regions that are more enriched for this genetic risk. Thalamus, frontal cortex, posterior cortex, and less in some other regions. And so this is really preliminary work because of the nature of where we got this gene expression data. A lot of it could be from just parts of the brain. This one is more complete, but came from a mouse. And so there's going to be a lot of better data coming in from human tissue that will integrate with genetics to gain deeper insights into the etiology of these conditions. More importantly is the biological inferences from individual genes. And so one of these genes is GRIN2A, which is a subunit of the NMDA receptor. There's a GWAS common variant seen in 35% of people that is right over the gene. And it gives you an odds ratio of risk increase of like 1.07. So it's not large amounts of risk. What we found in the sequencing study is that if you have a loss of function variant in that gene, a stop codon or a frameshift mutation, you have a 20-fold higher risk of schizophrenia. And so we have a regulatory modulation loss of function effect that confers substantial risk for schizophrenia. And so it kind of puts additional evidence in the glutamate hypothesis, the dysregulation of glutamate as a meaningful process that is one of the likely processes that lead to psychosis. Now, GRIN2A is especially interesting. It is part of the NMDA receptor. There's 2 GRIN1 units, and there's a GRIN2A and a B unit. And the GRIN1 units, there's 2 of them, they're always there. and the A and B unit switch in abundances over development. And what we see is that the GRIN2A unit linked to schizophrenia is not expressed in early development, and it switches on in postnatal development. And schizophrenia's onset is later on in development. And so we get a matching pattern from gene expression, as well as the genetic evidence and the epidemiological evidence, and with the glutamate hypothesis, pharmacological evidence with ketamine, that this whole pathway is likely interesting. Now, I mentioned GRIN2B as the other unit of the NMDA receptor, and GRIN2B is linked to autism. And so you get this nice switch. The composition is mostly B, switches on to A, and what we see there is that each one is linked to different neurodevelopmental and neuropsychiatric disorders. And then there's another gene, GRIA3, which is here, that is also linked to schizophrenia and is part of the AMPA receptor. It's an AMPA receptor subunit. And so not only do we see pathways and processes, but also individual genes that tell us quite a bit about the pathophysiology and the etiology, clues towards the etiology of schizophrenia. The other really important analyses I think that have come out in the last couple of years is how we compare across disorders. We can learn a lot by comparing across neuropsychiatric disorders and neurodevelopmental disorders. And in parallel, as we did a lot of the schizophrenia work, there are amazing papers in intellectual disability, autism, epilepsy, bipolar, as part of the international community, and we can use these data to learn more about schizophrenia. And so let's talk about the specificity. Let me get some water out. Okay. Specificity of individual genes for schizophrenia and intellectual disability. TRIO. It is a gene that's linked to schizophrenia. And on the top part of this gene, you can see the exons and introns are these stop and loss mutations that are linked, that we see in schizophrenia patients. There's 20 of them. Now, we can also plot mutations from patients with intellectual disability. And this is what it looks like. They all cluster in a singular domain of the gene, all in the middle in one functional protein domain. What is also important is that these are all missense variants. They're not loss of function. They're not stop codons. They actually change the structure of that domain. And so one gene, different mutations, different phenotype. And so these patterns are what we were beginning to see, and it gives us clues about what to do with these genes in functional models. And so this isn't the only gene like this. SETD1A, so this part is a bit complicated, but there's a loss of function part, PTD is loss of function, and there's a missense part. And if you look at SETD1A, the loss of function variants confer risk that they're more red in schizophrenia and intellectual disability. But in TRIO, calcium channel CACNA1G, and GRIN2A, the glutamate receptor, it's all stop codons and loss of function variants in schizophrenia, and they're missense in intellectual disability. Different mutations, different regions. And this is what it looks like in the NMDA receptor. Loss of function variants in schizophrenia, these are the missense variants. They're all in the pore of the NMDA receptor. And they're all alternating, they're opening the channel. Through functional studies, we know that now. And so one gene, one protein, different mutations, different phenotypes. And we see this a lot. There's a lot of genes that have shared patterns with neurodevelopment and different patterns also with neurodevelopment. And we're going to find loads of these genes guaranteed in the future. And there's a lot of avenues of analysis where we can combine them, contrast them, which is pretty exciting. Now, what is also true is that there's genes that are specific to schizophrenia. So I mentioned exportin 7. There's brain transcription factor SP4, AMPA receptor GRIA3. These have no signal in intellectual disability. They're only mutated, affected in patients with schizophrenia and broader neuropsychiatric disorders. So what we're seeing is genes that are unique to schizophrenia, shared with neurodevelopmental disorders, and shared in different ways that lead to different phenotypes. And this map is going to be incredibly valuable as we try to understand biology of these conditions. The final bit I don't want to spend too much time on is that we now have GWAS, the common variants, common small effect, and we have the rare variants. The question is, do they hit the same genes, or is it completely different biology? And the initial clues show that there are shared genes, GRIN2A and NMDA receptor. I mentioned that earlier. Common variant over the gene, loss of function variants confer risk. Brain transcription factor SP4, loss of function variant confers risk, common small effect variant right over the gene. And then there's two other more complicated examples where, if you look at this here, it's hard to know what the gene is, but there's something there. It's an important common variant locus. And if we look at the genes though, only one gene in each one have rare variant signal. And so we could look at these rare, more damaging variants and these common variants and find convergent biology. And so there's complementary approaches now to understand the biology across the genetic architecture of schizophrenia. And finally, how much does this actually matter in patients in terms of presenting and observing them in patients? And so we did a number of analyses to estimate this across a global collection. And the numbers that we reach are about 6 to 7% of patients will carry a loss of function variant that confer some risk for schizophrenia, so a protein coding variant. And if we break that number up, about 24%, 26% are de novo. The parents never had them. And the other 3/4 are inherited. Now, it seems like then there's a lot of inheritance, but actually the 25% of being de novo indicates that these are probably recent generation variants, that they appeared and they're slowly removed from the population because the effects are so large. So the key takeaway from this slide I want you to take is that these are rare. If you go to a clinic and you meet a patient with bipolar or schizophrenia, only a small fraction of them will have these variants. And that affects what we do with this knowledge in terms of understanding biology, approaches, patient identification, diagnoses. I think there's a lot of interesting work to be done with more detailed and more comprehensive collections. And finally, this here I use to show signal in rare variation. On the right side is loss of function variants. And there's a one line, which is if there's no signal. And so there's a lot of signal in the loss of function variant category. If we remove every gene I talked about today from that signal, in theory, if there's no other gene out there, it should drop to 1. However, I remove all the genes, it's still there. There are loads of genes to be found, hundreds of genes. We're at the beginning of the process of finding more genes for schizophrenia. And I think all of those will come together to give us a better picture of the genetic architecture. And so right now, there's active work to work on the phase 2 part of the schema analysis. And it's quite exciting. We had 7,000, we had 25,000, now we have 50,000. 50,000, 300,000 controls. This is not up to date. And so this is thanks to leaders in the global community, obviously in Boston, also in the UK, around the world that have come together to work on this problem simultaneously. And so in the next year or two, probably next year, we're gonna get a lot more genes for schizophrenia, doubling the case numbers, tripling the control numbers. It's gonna work. Now, there are certain patterns that we're seeing, and I'll highlight one of them. So in the initial analysis, there was a gene in the FDR less confident list that was particularly interesting, called DAGLA. And we're beginning to see a lot of signal in this gene in preliminary analyses. What is DAGLA? DAGLA produces 2-AG. It is a principal component enzyme in the endocannabinoid pathway. It produces the principal endocannabinoid that binds to the endocannabinoid receptor. And as you might know, THC in cannabis binds to the same receptor to produce their biological effects. And so what we're beginning to see, both in the initial analysis where there's clues and with more data, is that we're going to find a lot of interesting pathways, new ones that we never thought about. We know the glutamate pathway likely is important, but the endocannabinoid pathway, well, we'll see if that stands in the next couple of months. And we're going to get many clues like this that we'll have to piece together to understand what causes schizophrenia. Okay, so I'm going to jump to another study now. And this is the Finnish biobank study. And it's a pretty remarkable study that took 7 years to come together. And psychiatrists led by Arno Palotti, a leader in Finland, they went around 5 hospital districts in Finland and collected and met, well, psychiatric nurses drove to every one of those hospital areas. Each dot is a different patient, and they met and recruited 10,000 patients with psychosis in Finland. There's only 5 million people in Finland. That's a quarter of everybody with psychosis. And it's an incredible work. There's psychiatric nurses here, psychiatrists there, Arno's a psychiatrist, a clinician, and they all came together to put together this amazing data set. And there are patients with schizophrenia, bipolar, other psychosis, schizoaffective disorders, and psychotic depression. Not only that, but we have deep phenotyping. Finland has an amazing electronic health record system that spans decades. And so we can integrate information about other diagnoses as well. And this data set has genetic data, whole exome, whole gene, some whole genome, and also very detailed controls as well. And there's a lot of takeaways that I don't want to go into today. I want to focus on a few things. The first one is that we are beginning to find axes of severity that we didn't see before. And so if you look on the left-hand side there, this is the genetic enrichment plot I showed earlier. When there's a 1 there, that means there's no enrichment. And if it's above there, there's enrichment. And here we're separating in 2 groups: patients with cognitive impairment, patients without cognitive impairment. About 5% of patients have cognitive impairment in schizophrenia in population surveys. And we find that the loading of genetic risk is substantially higher in this group, much higher across the board. When we look at intellectual disability genes, we see that same massive enrichment, 4-fold in cases, much less in the remaining population of schizophrenia individuals. And finally, when we look at the genes that we discovered in schizophrenia, they're also enriched in this group. So just to highlight some of the numbers there, about 8% of patients will carry an intellectual disability gene in that group, compared to under 3% in the cognitive impairment group. So we're seeing axes of phenotype enrichment. I could show this for common variants too, in multiple phenotypic dimensions, but also obviously severity. Schizophrenia patients have just more genetic loading than any other diagnosis. People with more genetic loading are more likely to be treatment resistant, clozapine prescribed, and they're more likely to be under kind of national support, disability pension, or kind of long-term care. And also the onset is about 2 to 3 years early. And so we're getting patterns across the board. This is a taste of kind of the deep kind of analyses we can do with genetic data and phenotypic data. And so what we can also do is find patients and combine data sets and see what the individual patients present with. So SETD1A, we now know of a lot of patients with schizophrenia with this variant. We also know, we also know that it leads to intellectual disability. So there's patients also with intellectual disability with this variant. And we could begin to see what their diagnoses are. Surprisingly, not that many of them have learning difficulty. We went back and looked at these patients, and the vast majority have schizophrenia. Some had bipolar, some have very complicated diagnoses, but predominantly it's schizophrenia, despite our prior that led to more severe neurodevelopmental disorders. And then when we looked at these patients, there's 33 now in the global population. We found that 12 were at the same base pair. There's a 2 base deletion that we see in 12 of them. 30% of these patients have this 2 base deletion. It's remarkable that we see this in schizophrenia, in broader psychosis, and intellectual disability. And so we could begin to find point mutants that are seen in large numbers of people that warrant additional functional study. There's genes like ACAP11, where a genetic study in bipolar indicated it's likely linked to bipolar, and our study in schizophrenia also found evidence of that too. So it's a gene for schizophrenia and bipolar. When we look at the patients in the Finnish study, they have schizoaffective disorder, bipolar, schizophrenia. It's a mixed bag, which recapitulates what we saw in the larger study. There's genes like MAGE2, where we only see the signal in schizophrenia, none in intellectual disability. Vast majority of patients, they all have schizophrenia. And so there's more schizophrenia-specific signal as well. And so what we can do, just taking the last 2 vignettes, is we can gain properties and we can learn properties of what these genetic risk factors do with deeper psychiatric phenotyping. And we can find patients for which we go in even more deeper detail and see their vast kind of complex psychiatric presentation over pretty much a lifetime. And so it's a powerful tool that we could begin to tap into to understand the biology of these conditions. And so just to summarize this part of the talk and wrap up, this vignette, is that the identification of rare large effect variants complement now a full understanding of the genetic architecture that will help us elucidate biological insights and heterogeneity within these conditions. There needs to be global studies as part of the global community to push these forward. But we also need more detailed studies like the super study, the Finnish study, that'll help us find these patients and study them in more detail. And so I think there's a number of really interesting approaches that will emerge and opportunities that build on the work, some of this kind of work in the future. And one thing I want to highlight is that, I presented some of this work here, which is mainly in schizophrenia, but I've been fortunate enough to work with a number of investigators across disorders in autism, epilepsy, intellectual disability, and functional studies. And there's no reason why this is limited to schizophrenia. It isn't. And so these approaches should be applied across the board, across neuropsychiatric disorders. Some will be easier than others for a number of reasons, but there's a generalizable tool set that we can use in genetics. And so I kind of go back to the slide quite a bit. Genetics supports, I think, it's going to be a major tool in supporting the state-of-the-art research in medicine. And you can imagine how these insights can influence studies in model organisms, epidemiological studies, imaging studies, looking at brain banks, clinical trials, and diagnosis. Some will come earlier than others, but hopefully they'll lead to better treatments. And so for this part of the talk, I want to thank the global community. I've been part of this community for 7-8 years, and it's 10 years actually. Man, time flies. But then it's really an international community with a lot of people in Boston, in Europe, in the United States, funded by the NIMH. There's a lot of people that come in and work together to do this. And it is a resource that we should tap into as we build out more genetics research, in New York and also at Columbia. And so with the final 8 minutes, something like that, I'm going to talk about what we're interested in doing. And so we really want to keep pushing the discovery of genes for neuropsychiatric disorders. And we're part of these global collaborations that have done this, and we want to keep contributing to it. And we're interested in expanding our scope to a wider range of diagnoses and developing new methods for new genomic technology. So I mentioned the next wave of the schema analysis, but Thomas Lehner, the scientific director at the Genome Center, we've been brainstorming about ways to build out collaborations using his, rich connections and interests in genetic technologies to kind of find new avenues. One of these is whole genome sequencing. And so let me explain this really briefly. This here is a composition chart of all the elements in the human genome. We've been mainly focusing on the 2% part of it, the exome. There's a lot of it around there that we don't study. Now, were we right to study the exome? Yes, it's much easier. Genes are easy, relatively. However, the numbers are reaching a saturation point where we now have a couple of 100,000 genomes sequenced in the world. And so there is no reason why we don't begin to look for patterns in the other 98% of the genome. And those are some of the ideas that we want to push forward in the near future. We are also interested in characterizing in more detail clinical outcomes of common and rare genetic risks by studying clinical collections like the Finnish project and also population biobanks. There's a lot of data now that's been committed by national governments where we can begin to think more about diagnoses, genetic risk, overlapping phenotypes, multimodal data, and so forth. And some of the great ones are All of Us, which is emerging, the UK Biobank, the Finnish Biobank. There's a long list now. And the expertise we've gained from analyzing schizophrenia, we should apply across the board. And one of the key goals in the last couple of months is to build that engine here at Columbia and using the resources at New York Genome Center to leverage cloud computing, local cluster computing, computer scientists to build an engine so we can analyze these data at scale using new approaches like machine learning and deep learning. The third angle is functional genomics. We have genes, we don't know what they do. And so there needs to be collaborations across the board to use these models to understand the functional consequences of genetic risk. So where do geneticists come in? there are now approaches that extend nucleic acid research where we could generate large amounts of data using biological systems. Some of these include single-cell RNA-seq, different forms of transcriptomic measurements, also multimodal imaging. So there's a lot of different approaches once we generate these systems. And so we've been brainstorming cellular systems, other approaches that we can leverage our sequencing expertise to then get us a bit closer to understand the functional consequences of what these genetic factors do. So one project I want to highlight that's brought into place by Thomas, who's right there, and Colleen Karam, the Associate Scientific Director of the Genome Center, is what we call a lens into genetic architecture of schizophrenia using deep whole genome sequencing of severe patients. And to do this, we're leveraging a collaboration that Thomas has had for many years. 65,000 patients that have been recruited and banked in terms of stem cells as well as blood samples. And we will use that rich biobank to find 3,000 of the most severe patients. And we'll use new, cheaper sequencing technologies. And what we'll do with those sequences is we'll combine it with everything that's been sequenced before in Schema, in other projects that have been funded to do a meta-analysis using deep whole genome sequencing data to extend the genetic architecture of psychosis. And what is crucial is that there are already preserved cell lines for these patients. And so we'll use those hopefully to do pilot functional studies as well. And this is already funded through great work by Thomas and his team. So really excited to push that forward. And it's happening now, and hopefully we'll push that forward very soon. Another example project is how we use machine learning and deep learning to take DEXA scans, X-ray scans, and do extraction of human phenotypes. This was a foray 2 years ago when we were trying to test how effective deep learning is to look at medical imaging. And we were able to extract skeletal features, many of them, just by using scalable deep learning technology. And so we applied this to human skeletons, and then we ran genetic studies on 30,000 people. And what we found were genes and factors that linked to skeletal disorders, that led to osteoarthritis, genes that regulate how the knee forms, how long your arms are. And these same genes are also related to why we're different to primates, to monkeys, because we have different lengths of arms. And so this paper is now in revision, and we're excited to apply these approaches into neurodevelopmental and neuropsychiatric phenotypes. Deep learning approaches to look at vast amounts of EHR data, as well as questionnaire data and form data to kind of push forward more complex genetic studies. So that's an area that builds on the biobank work that has been yielding results, and we're just going to keep pushing that further forward. And then one angle I think I'm going to highlight here, it feels more concrete, is work at Mind, the Mind division. And there's an amazing postmortem brain collection led by the investigators here, John, Mara, Hanga, Andrew, all of these investigators who have built over decades, including Barbara Stanley. There's just a lot of people that have been working on this over 20, 30 years of postmortem brain tissue, of people who've committed suicide. And there's a lot of important work that's already been done to look at the neurobiology of suicide, including molecular signatures. And so one proposal we submitted that is in review right now is we want to whole genome sequence 200 patients, leverage Mara's expertise in multimodal measurements in single cells, and then integrate the two to see which clusters of brain cells are different in patients and controls, and then map it to the genes that we've linked in suicide to refine those patterns and kind of pinpoint meaningful genes. And this will require overlaying many types of data together, focus on genetic and functional data to better understand the causes of suicide. And so these are some preliminary projects, some that are happening right now, and some that will also go further, hopefully soon, that we could begin to use genomic expertise in a range of our areas of research here. And so I want to end here. I want to thank a lot of people that have helped me onboard at Columbia, Murray, Anthony, and Angie at Mind, who have been helpful in kind of the administrative processes, Blair, Amy, Iris, Larry, Mel, Kai, Simon, people that really helped me kind of onboard and get going here. And obviously, I mentioned Thomas and Colleen here many times, but they've been really helpful in orienting me about different opportunities in New York, at Columbia, that we could push forward. And obviously, John, Melanie, and people in mine in the mental health data science division who have helped me also kind of orient myself of the structure in our department and find opportunities there. And finally, we're looking to expand our team and collaborations and strengthening our ties with Columbia and the New York community, looking for postdocs, computational research associates, graduate students, residents. There's a lot of different angles that we could take this that require different skill sets. And obviously, I would love to collaborate more with different faculty here. And so it's only been 5 months, but I'm sure we'll find more chances to talk in the near future. And so I'll stop here, and I'm happy to take any questions.